All right, good morning, everybody. We might look to kick this off. I can see the numbers are starting to settle down there in terms of participants. We've got another great turnout for today, which is no surprise given the speaker that we have today. Before I begin, I would like to acknowledge the Wurundjeri people of the Kulin Nations, traditional custodians of the land on which we are based, and I wish to pay my respect to elders past, present and emerging. It gives me great pleasure today to introduce Professor Mark Dawson. I'm sure that Mark is known to many of you, if not most of you. Mark is certainly recognised as one of the leading clinical scientists, clinician scientists we have here in Australia, and has a number of positions here within the precinct. He heads the Translational Haematology program, the group leader in cancer epigenetics, and indeed is a consultant haematologist for the Department of Haematology. He is also an active member, a relatively new member of the centre, which some of you may not be aware of. And we're absolutely delighted to have him presenting here today. Mark's research interests have really focused on epigenetic regulation of cancers, ranging from initiation, maintenance and cancer progression. And I'm looking very much forward to seeing what Mark is going to present today on targeting the epigenome in cancer.
Thank you, Sean, and thanks everyone for spending your Wednesday morning with me. So as Sean just alluded to, I'm a clinician scientist and work at the Peter MacCallum Cancer Centre, but also am an active participant and member of the Centre for Cancer Research at the University of Melbourne. So I'm very grateful to be part of Sean's faculty there. My work aims to try and make new discoveries that we ultimately take into the clinical arena to improve the diagnosis management of patients. And really, what we do is try and test this in the clinical arena, learn from that, and then reiterate this process. I want to give you a summary today of the kind of major areas of interest for my lab, and then I'll present 3 stories highlighting these. Really underpinning the engine room of discovery for the lab is broadly genetic screens. These take the form of both RNAi and CRISPR screens. And today I'll talk to you about some of our work using both these methodologies. Another major area of interest for the lab is really understanding the origins of cancer. We have now known for a while that preceding malignancy, there is a clonal outgrowth within what is essentially looking at the tissue. This has probably been best studied in the hematopoietic system and is known as clonal hematopoiesis. And my lab is really mainly interested in understanding the consequences of clonal hematopoiesis, the underpinnings of this in the context of both infection and inflammation. And hopefully in the next few months, you'll see some new work on this from our lab. Another thing that I'm going to talk to you today about is plasticity and transcriptional plasticity. We believe that this plays a major role in directing cell fate decisions in cancer and really underpins quite a lot of the therapeutic resistance we see associated with various malignancies progressing on all types of conventional targeted and immunotherapies. I work very closely with Sarah Jane Dawson, and I know that this audience probably heard from her a couple of weeks ago. And together we really try and understand tumor heterogeneity, primarily in this case through a genetic lens using circulating tumor DNA. More recently, my laboratory has been very interested in understanding the interplay between the epigenome and how it may be manipulated to increase immune surveillance. And again, today I will explain some of our recent work in this area. Finally, my lab really has a major interest and understanding or interest in chemical biology. And we have over many years now worked with a number of medicinal chemists around the world to try and improve and innovate and develop new small molecule inhibitors for epigenetic regulators, but also understand better ways in which we may be able to monitor their preclinical and clinical efficacy. So let's get into it. I guess most of my lab, about half of my lab, really works on hematological malignancies. And the malignancy that we're interested in understanding is acute myeloid leukemia. This certainly is the major component of my clinical interest, and largely now my clinical practice is confined to treating patients with acute leukemia, both ALL and AML. The origin of this malignancy has been really well studied, and what we understand here is that a leukemogenic mutation either occurs in the hematopoietic stem cell or reconfers self-renewal properties to a hematopoietic stem, a progenitor cell, and immortalizes this cell, and consequently, over a period of time, with additional mutations, leads to the full-blown malignancy. Leukemia was one of the first malignancies also described to have a similar hierarchical structure to normal tissue. And at its apex is the cancer stem cell, which is called a leukemia stem cell. The vast majority of leukemic cells are incapable of ongoing malignant self-renewal, encapsulate the malignancy if it was transplanted into a susceptible recipient. How then do we think about treating this cancer? This remains one of the poorest prognosis cancers we have. We cure with conventional therapy only about 30% of these patients. That's not to say that chemotherapy is not effective. In fact, chemotherapy alone puts up to 80% of our patients into a complete remission. That remission is not sustained, and ultimately the relapse happens primarily because we're unable to eradicate the leukemia stem cell. But really, how do we understand chemotherapy, or in fact therapeutic resistance? Largely, we think about it in this way, through a genetic lens. We believe that chemotherapy, which includes DNA damaging agents, induces mutations that results in genetic clonal evolution. This then confers upon the cell these new mutations a survival or proliferative advantage, and then this becomes the dominant population. And this really has been reinforced through imagery like this, the classic fish plot. But for some time, we have wondered whether this might be the dominant force by which cancer evolution in the context of any pressure, but particularly therapeutic pressure, happens. Several years ago, my lab, when we were studying the principles that underpin resistance to a targeted therapy, in this case an epigenetic therapy, published this paper. And here I really want to emphasize two major principles. Firstly, this was one of the first examples by which we showed that resistance occurs from cells markedly enriched for leukemia stem cell potential. But the second major thing that we found is that when we looked at the fully resistant cells increased for leukemia stem cell potential and compared it to the drug-naive cells, there was absolutely no evidence for gene. Although this period of resistance had taken up to a year to develop, there were no new mutations. And essentially what had happened is that the leukemia stem cells had rewired themselves to evade the therapeutic pressure and use an alternative transcriptional pathway. Now, this was done in a model organism and with an experimental therapy. So the real clinical relevance of this remained largely unknown for a couple of years until this paper from John Dick's lab was published. And here, what Liran Slush and John Dick showed was essentially when they look at patient samples now treated with conventional therapy and assess them both genetically and functionally at baseline, at diagnosis, at the time of remission, and then subsequently at relapse, what they found were identical principles. Here again, they showed very clearly that relapse from cells markedly enriched for leukemia stem cell potential. And really importantly, in none of the cases that they identified, even though resistance took months, if not years to develop, did the resistant cells have any new coding mutations that could account for the resistance they saw emerging from the leukemia stem cells. In fact, now we have gone back and had a look at this in far more detail. And essentially what you see on this slide is all of the examples to date where there has been paired samples from diagnosis and relapse and whole genome or whole exome sequencing published for patients with acute myeloid leukemia. And to summarize this, wherever you do not see a red bar, there is no evidence of genetic evolution at the time of relapse. And in fact, many, up to 40% in some cases, of the now look like this, which is, it suggests that what you start with is really what you end up with, and primarily the resistance occurs in the absence of genetic evolution. So that brings us to essentially what is one of the major focuses of my lab. Right? What is the driving force for cancer evolution? Is it really Darwinian? Is it really survival of the fittest? Or did Lamarck have a fair example here, you know, of environmental adaptation? And in part, you know, understanding this has been clouded by some of the studies that have been done because of sequencing depth, whereby, you know, imagery such as this fish plot suggest a sequential genetic evolution model. Here, what we imagine to happen is that these cells serve as the origin for cancer, and as a consequence of metabolic, hypoxic, nutritional pressure, mutations eventually develop, giving rise to this tumor. Then in the context of therapeutic pressure, you know, DNA damaging agents induce new mutations, and then you see this brown cell appearing. Now, when studied at single cell resolution, what we're beginning to see is really quite a different picture, which is that all of this heterogeneity exists right at the get-go, and it's present in the cancer stem cells. Every single mutation that you see on here is already present to start with. And what happens is that the fittest cell in a particular pressure environment is what dominates. And as this changes over the course of a lifetime of the individual, and certainly in the lifetime of a tumor, that representation also changes accordingly. And hopefully over the next few years, you'll see some experimental evidence from our lab and the others confirming this process. Brings us to the point of, well, are leukemia stem cells, if we consider the major concepts of non-genetic evolution. So here, you know, these cells are getting resistant to therapeutic challenge. And as a consequence, and this is happening in the absence of genetic evolution. So are these leukemia stem cells primed or plastic? That's the next major question we really want to answer. What does that mean? In the consequence of prime, they either have the right metabolic transcriptional program right at the start, and all the therapeutic pressure does is select for them. In the situation where they are plastic, you know, this advantage to survive therapy is not inherent, but is adaptable. That is, the cell in the context of the therapy rapidly changes and is now able to adapt as a consequence of it being a stem cell. And as a result of that leads to the resistant population. In both cases here, the destination is identical, but the route to resistance is very different. In this case, what we want to do, and the consequence of that is that, you know, treatment to stop resistance happening is very different. In this case here, we want to target whatever this transcriptional program or metabolic signature is that results in priming for resistance. In this case here, we want to target, you know, the adaptive process, the rewiring that leads to resistance. And my strong belief is that these things are not mutually exclusive. And therefore we need to understand the major principles that underpin these processes. So in terms of adaptation, we've recently published some findings showing that leukemia stem cells are able to adapt to therapeutic resistance by using a process called enhancer switching. Enhancer switching is a normal process. Here is an example to try and illustrate this. So here what you have are myeloid progenitors, B cells and T cells. By and large, they all express MYC, which is the prototypical oncogene required for normal homeostasis as well. And you know, the growth and survival of many cells. MYC is expressed to a similar level in all of these lineages. However, the control of MYC varies substantially. In myeloid cells, it's mainly using this enhancer. In lymphoid cells, it's either using this or this, depending on whether it is a B or a T cell. And similarly, a cancer stem cell has all of these opportunities available to it. So as we shut off key oncogenes such as MYC, it rewires itself to throw up new enhancers to be able to reinstate the expression of these key oncogenes, such as MYC, BCL2, etc. All of the major oncogenes that many of us have studied over the years. As a consequence of throwing up new enhancers, what we see is redistribution of the major components of the transcriptional machinery, including coactivators such as BRD4. So BRD4 moves from a pre-existing enhancer to a new enhancer to allow that to become functional. Why is this important? This process of adaptation occurs at the time of minimal residual disease, when the cells are rewiring itself to try and find a new way to survive in this hostile environment. And thus far, epigenetic therapies have really mainly been studied in the context of overtly relapsed and refractory cancers. And let's be fair, they have had a modest, if not poor outcome in these diseases. But what we're proposing is to start to use epigenetic therapies at the time as maintenance therapy, when there is minimal disease, and these cells are adapting. And consequently, we've started this national clinical trial in acute myeloid leukemia, where we're repurposing this drug, not to use it up front, but at the time where this adaptation is happening, to disengage and to disempower this adaptation process. This study will take place over the next few years, and we'll hopefully understand whether maintenance therapies in the context of acute myeloid leukemia may change what is currently a dismal outcome. As I said to you, this process is not mutually exclusive. So there is definitely, transcriptional programs within leukemia stem cells that enable them to survive therapeutic pressure. So we really wanted to understand this in greater detail. And to do this, what we set out to do was a large genetic screen as a small molecule. And this is work that was led by Laura McPherson in the lab that started in about 2015 and has only recently been published. So she set out initially to look for novel therapeutic targets in leukemia stem cells, initially using an RNAi screen. Why we chose RNAi over CRISPR, we can talk about, but I think this is an important strategy, particularly when it comes to epigenetic regulators. But as you know, regardless of what your genetic screening approach is, what you find is essentially your gene of interest. It doesn't tell you how to target this gene. Because here is an example of an epigenetic regulator. There are a number of potential drug targets. There is the catalytic domain. There is the domains that help bind to DNA. There are the protein-protein interaction domains. All of these are viable therapeutic targets. So to find the precise therapeutic target, we adapted a method, a good friend and colleague, Chris Farkoch, called CRISPR domain screening. Here, what this methodology aims to identify is exactly the domain that results in this being a good therapeutic target. And then in collaboration with medicinal chemists, in this case, it was the CTX-CRC and Jonathan Bale at MIPS, we sought to identify new small molecule inhibitors of this target and then take them back into the preclinical setting to see if the small molecule can essentially phenocopy the genetics. And this is the work of Laura that I'm about to present. So the first thing to show you is the fact that the genetic screen was incredibly reproducible. The novel targets that we identified can be seen here on this PC8 plot. And essentially what this shows you is two screens done 3 months apart, highlighted and blue. The screens identified unique targets to bulk leukemic blasts, but also unique targets to leukemia stem cells. It's fair to say that the majority of targets were equally shared between both. What did we find and how did it compare to what had been found before? Acute myeloid leukemia served as the screening ground for lots of previous therapies, including many epigenetic therapies, resulting in compounds that have entered into clinical trials. And this is what we see when we look at those targets. When we knock down EZH2, LSD1, PRMT5, DOT1L, BRD4, all of them are exceptional at eradicating the bulk leukemic blasts, but none of them are capable of getting rid of the leukemia stem cells. In contrast, what we did find was a new potential therapeutic target, a missed acetyl transferase called HBO1, which was very important in eradicating, but equally important in eradicating leukemic blasts, as shown here by this validation. As you can see here, this is RNAi. So we have substantial decrease in functional protein, but not complete eradication. And I think that this is important because this probably parallels the best clinical activity that we can get with small molecule inhibitors. Then, as I said to you, we went on to identify which is the precise domain against which we needed to develop a target. And using CRISPR domain screening, this very clearly ended up being the missed acetyl transferase domain, which bore the catalytic activity of the drug. To really emphasize the fact that it was the missed acetyl transferase domain, we did this experiment. Here what we did was knock out HBO1 and rescued it with the guide-resistant wild type or a guide-resistant construct that contained a single point mutation that abrogated the catalytic activity of the drug. And what you very clearly see here is that the wild type construct fully rescues the phenotype, whereas loss of catalytic activity was unable to rescue the phenotype. This is probably the best example of showing you that this is the right target to which to build a drug. Now, this is all our genetic approaches, but is it really required for leukemia stem cell maintenance? And the major phenotypes that we found were an increase in apoptosis, a cell cycle arrest, and a differentiation phenotype. But all of that is in vitro, and leukemia stem cell activity is done in vivo. So here we built several mouse models of disease. And here I'm showing you an example where we combine the two most common mutations in acute myeloid mutation, NPM1, nuclear phosphate mutation, and a FLT3 gain of function mutation in this tyrosine kinase. In both these cases, mutations were knocked into the endogenous locus of the mouse, so physiological expression, and the mouse expressed both of these concurrently. Then using inducible RNAi, what we were able to clearly show is that if you knock down HBO1, you completely abrogate the detrimental effects of these mutations. Mice that expressed the non-targeting construct quickly succumbed to the disease, yet the only mouse that succumbed to the disease in the HBO1 knockout was one in which there was, for some reason, an inability of the RNAi to work, as shown here. HBO1 is expressed, if not higher, than the parental cells. We also showed this in Cre-Lox mice and in CRISPR models as well. So, as I said to you, the major phenotypes that we saw is an increase in a cell cycle arrest, an increase in apoptosis, and a prominent differentiation of these leukemia stem cells. What is the molecular mechanism underpinning this? HBO1 is a transcriptional activator, and it acetylates a key histone residue called H3K14 acetylation. And when we look at the top 20 genes downregulated following loss of HBO1, top 25 genes, many of these are known genes important for the maintenance of leukemia stem cells, particularly those that belong to the HOX cluster. Interestingly, when we look at chromatin, what we see is that these genes have H3K14 acetylation dispersed over the entire coding region of these genes. And here is RNA polymerase massively low these, showing that they are highly expressed. In fact, when we look at these genes that HBO1 regulates, as shown here in green, the majority of them are the highest expressed genes in a cellular phenotype. So I'm not going to be able to take you through the entire molecular mechanism. Instead, I'll show you what we found in this schematic. Using proteomics, what we were able to show is that HBO1 associates with MLL1 and consequently is recruited by wild-type MLL or indeed MLL fusions because it binds to the N-terminus of MLL to chromatin, to certain target genes. And here it acetylates lysine 14 on histone H3. This histone modification, which is conserved from yeast to mammals, then serves as the beacon or landing platform for chromatin remodeling complexes that contain proteins that have a bromide recognize this histone modification. They then subsequently become recruited and bound to H3K14 acetylation on these target genes. And components of the SWI/SNF and ISWI chromatin remodeling complex rapidly remodel chromatin in evicting nucleosomes, enabling the rapid transition of RNA polymerase. This increased processivity of RNA polymerase leads to the high expression of these target genes that ultimately drives the maintenance of the leukemia stem cells. So I want to move on now and talk about what we did next. Targeting acetyltransferases selectively has really been fraught with problems for nearly 2 decades. Only recently have we been able to develop selective small molecule inhibitors to acetyltransferases. And this is potentially the second example of such a selective small molecule inhibitors to CDP and P300 developed by AbbVie. And this really highlights, the leading medicinal chemistry we have here in Melbourne, because this was an incredibly difficult feat. Here I'm showing you here is a prototypical small molecule inhibitor called WM3835, which binds very selectively into the missed acetyltransferase pocket of the missed acetyltransferase family, which includes HBO1. And here we were able to use structure-based design to increase the potency and build out this small molecule to make selective contacts that are only present in HBO1, increasing the potency of this small molecule to HBO1. Now, taking this back into the preclinical setting, what we can see here is that in every example where there is a genetic susceptibility to loss of HBO1, the small molecule inhibitor works equally well. Where it has no phenotype on the growth and survival of these cells, the small molecule inhibitor does not either. As I showed you previously, loss of HBO1 leads to the loss of H3K14 acetylation. And again here, that's exactly what we see with the small molecule inhibitor. And it reduces the expression of these key HOX genes, both in mouse and human cells, required for the maintenance of these cells. Again, it induces a cell cycle arrest and an increase in apoptosis and a prominent differentiation, phenocopying the effects that we had seen with genetic manipulation of HBO1. And whilst this compound was not able to be used in vivo, we were able to show ex vivo that it inhibits the activity of leukemia stem cells in patient samples. So I'm going to summarize this work, which was recently published, by showing you that we used a novel isogenic model of AML blast and leukemia stem cells to uncover commonalities and differences between the populations. Using RNAi, we identified HBO1 as a novel target. Using CRISPR domain screening, we showed that the major target to which to build a drug was the HAT domain of HBO1. We worked out the cellular and molecular mechanism by which HBO1 maintains leukemia stem cells in vitro and in vivo. And now we have the knowledge to develop more potent small molecules against HBO1. And in fact, this has been happening. And we're really encouraged by the fact that small molecule inhibition of HBO1 is likely to enter the clinical arena in the next 12 to 24 months with a bit of work. I now want to switch focus and talk to you about immune evasion and the role of the epigenome in this process. And this is work that has been led by a postdoc in the lab, Marianne Burr, who's recently moved on to start her lab at the ANU. She initially started by looking at this key indicator called PDL1 and wanted to identify, major components of the cellular machinery that regulated the expression of PDL1 on the cell surface. Now, this paper was published several years ago, so I'm not going to go through it, but essentially what we found in this manuscript was a previously unannotated and uncharacterized gene called CMTM6. CMTM6 turns out to be a master regulator of PDL1, not through transcriptional regulation, but instead what it does is that it regulates the endocytic recycling of PDL1. PDL1 transitions the secretory pathway after being transcribed and translated to reach the cell surface absolutely fine. But here it meets CMTM6. And PD-L1 is rapidly recycled. Endocytic recycling is a key component of proteins expressed at the cell machinery, and in this case, CMTM6 facilitates this process. The absence of CMTM6 is no longer recycled and is in fact shunted to the lysosome for degradation. And based on this work, we founded a small molecule, a new Australian company to try and look for small molecule inhibitors of this key regulation. But today, what I'm going to talk to you about is what happens if antigen is not presented in the first place. You know, there are a number of examples where lack of antigen presentation turns out to be a resistance mechanism to key immunotherapies, but also a major way by which cancers evade the immune system. So again, here what we took was a genome-wide CRISPR-based screen. Using 220,000 guide RNAs, we started with cells that failed to express class 1 on the cell surface. We knew that all of the coding genes for the class 1 antigen processing and presentation machinery were intact. So this wasn't as a result of genetic mutations. In fact, the vast majority of that antigen fails to be present on the cell surface in cancer is not due to genetic mutation. Only about 20% of resistance mechanisms to immunotherapies due to lack of antigen presentation is actually caused by mutations in genes. So we knew that this was a non-genetic mechanism. So using CRISPR, we then enriched for cells that subsequently presented antigen on the cell surface. And after multiple rounds, we're able to purify these cells. Here's an example of how this works. So this is what the cells look like prior to the CRISPR screen, and this is once they are now presenting antigen on the cell surface. What did we find? Well, every single dot above this line 4 here, which is where we called our significance line, are epigenetic regulators. And many of them belong to the same complex, which is the evolutionary conserved PRC2 complex. The key components of which are SUS12, EED, and the target is MTF2. Here's exactly how this works, and here's some of the validation. So here are the cells that are failing to present antigen on the cell surface. After taking out a key PRC2 member, in this case EED, we get upregulation of MHC1 on the cell surface. We know that PRC2 is necessary for this process because if we recomplement with wild-type cDNA, we can repress antigen presentation on the cell surface again. And here's the biochemistry to show you this. So again here, when we delete EED, we lose H3K23 trimethylation, a key histone modification associated with transcriptional repression. This results in MHC1 expression. If we are able to complement this with wild-type EED, we induce repression again, and we lose expression on the cell surface. We went through a number of processes identifying the key components involved in this process that I don't have time to go through today. What we then went on to do was ask, what are the major cancers that this transcriptional repression is likely to be responsible for? Looking at both human data as well as cell line databases. And what we see consistently at the end of this spectrum, with the lowest expression of the antigen processing and presentation machinery, as is encompassed here, are small cell lung cancer and neuroblastoma. And there are many other cancers in this part of the spectrum as well. Is PRC2 required for all of these cancers? The short answer is yes. So here are representative examples of small cell lung cancer, neuroblastoma, Merkel cell cancer, and erythroid leukemias, all of which, when we take out key members of the PRC2 complex, we increase antigen presentation on the cell surface. How is this done? This is through a coordinated transcriptional upregulation required for antigen presentation, for the peptide loading complex, and the key transcription factors associated with this, and also the immunoproteasome. So what I want you to take home from this slide is that all of these genes are coordinately regulated by the PRC2 complex. To show the relevance in vivo, we collaborated with Kate Sutherland, who had developed a wonderful model of small cell lung cancer, recapitulating the genetics that we see in human disease. Human small cell lung cancer ubiquitously inactivates P53 and RB biallelically, and as does this cancer. and this cancer, as there's this mouse model. And this mouse develops small cell lung cancer, which very much like the human disease, fails to express MHC class 1 on the cell surface. This is one of the examples of why non-small cell lung cancer is the poster child for immunotherapies, whereas small cell lung cancer, which is a translational burden, is not as responsive to immunotherapies. Importantly here, when we inhibit the PRC2 complex, we can increase antigen presentation on the cell surface. And as shown previously in human cells, this is a consequence of transcriptional upregulation of the entire antigen processing and presentation machinery. How does this work in terms of functional consequences? So here we used a number of in vitro and in vivo models to demonstrate this, and I'm going to show you one example of each. Here we took small cell lung cancer from this mouse model and expressed over albumin, which is then subsequently expressed on the cell surface once the competent antigen processing and presentation machinery is unleashed. And then we used OT1 T cells that specifically recognize over albumin presented via class 1 to see how it can eradicate these small cell lung cancer cells in a co-culture system. This here expression of inflammatory cytokines, interferon, for instance, by itself is not able to drive this process of T cell activation and tumor eradication. However, inhibition with EZH2, either by itself or in combination with interferon, very rapidly gets rid of these T cells, of these tumor cells due to T cell activation. This then led us to do an experiment to understand how immunogenic or lack of immunogenicity these tumor cells have. So here what we did was we transplanted the small cell lung cancer derived from C57BL6 into BALB/c mice, expecting that these would be rejected. But they were not rejected, in fact grew equally well in the BALB/c background, a genetically diverse host, than in the syngeneic background. This, in many ways, reminded us and the ability to evade and cross MHC class 1 boundaries was entirely dependent on PRC2, because if we take out members of the PRC2 complex, these tumors are rapidly rejected and none of these mice die of lung cancer. So as I said to you, it reminded us of transmissible tumors. Now, there are 3 known examples of transmissible tumors in nature, those that occur in marine animals, particularly clams, and there are several examples in various species of clams. Then there is the transmissible tumor that has been in continuous perpetuation for at least 10,000, if not potentially even up to 30,000 years, which is the sexually transmitted tumor of dogs. And then there is the tumor that is closer to home, which has been in continuous perpetuation for between 30 and 40 years. And that is the transmissible tumor that is affecting the population. Now, this tumor also fails to express antigen on the cell surface. But remarkably, if we inhibit the PRC2 complex in these cells, we're able to increase MHC class 1 presentation as a consequence of transcriptional upregulation of the entire antigen processing and presentation machinery. So what is, the clinical relevance of this? And here I just want to show you a case vignette of a patient who presented initially with an adenocarcinoma of the lung. She had an EGFR mutation and was treated with erlotinib to inhibit EGFR. And as is increasingly being recognized, despite the fact that she achieved a durable remission for over a year, she eventually relapsed. And in the consequence of relapse, underwent neuroendocrine transformation. That is, her adenocarcinoma became a small cell lung cancer. This is an infrequent occurrence in the context of potent targeted therapies. And what I want you to see here is that whilst the adenocarcinoma expressed MHC class 1, the small cell lung cancer component did not. She was subsequently tried to be salvaged with both chemotherapy as well as immunotherapy. And unfortunately, none of this was successful, and she eventually succumbed to the disease. And again, during the cascade examination of tumors from multiple sites, what we were clearly able to see is that the small cell component that probably ultimately led to her demise was class 1 null, whereas the adenocarcinoma continued to express class 1 on the cell surface, in part explaining why the small cell lung cancer was completely refractory to the immunotherapy. How does this work at the molecular level? Well, both in human and mouse cells, the PCS controls a number of genes. But when we look at the genes that are coordinately controlled by inflammatory cytokines in the PRC2 complex, well, the major components of those include all of the members of the antigen processing and presentation machinery. And the reason this happens is that the cell of origin seems to matter greatly here. In tumors that fail to express antigen at the cell surface, their cell of origin has bivalent chromatin modifications. Here it contains both a repressive chromatin modification as well as an activating chromatin modification. And these genes are poised but not transcriptionally activated. And here is an example of many of the human cancers that fail to express class 1 on the cell surface. This bivalent poising of antigen processing and presentation is not just true in human cancers, but also occurs in mouse cancers. And indeed, actually, we went back and did this in Tasmanian devil facial cells and saw exactly the same process. Why is this important? Well, as I said to you, the cell of origin seems to matter greatly. And tumors that arise as a consequence of arising in cells that have multipotent potential, or those that come from neuroprogenitor cells, such as the Tasmanian devil tumor cells, which come from a schwannoma, and certain melanomas, Merkel cell cancers, etc., also have a neurocrest origin. Many of these with neuroendocrine origin have poisoning of the transcriptional machinery. However, in tumors that come from the mesoderm or endoderm, here class 1 is resolved and this bivalency is completely resolved, and they express antigen on the cell surface just fine. So I'm going to summarize this part of my talk and show you here that we, in this paper, showed the PRC2 complex coordinates the developmental expression of genes involved in antigen processing. This repression not only represses class 1, but also restrains cytokine activation of these genes. The genes that are marked by bivalent chromatin in pluripotent cells serve as the cell of origin for many of these tumors. This developmental process is important because it provides these cells with an immune privileged environment. But however, when a cancer arises in this process, it really co-opts the PRC2 mediated bivalency to evade immune surveillance, even in the context of a high mutation burden. I show you that this process is evolutionarily conserved and is likely to contribute to transmissible tumors. And the key challenge for us ahead is how to selectively manipulate this process, because the answer has important implications for both cancer and autoimmunity. In the last 5 minutes of the talk, I just want to talk to you about some of our recent work looking at new epigenetic regulators. This has really been a new small molecule epigenetic regulators. This has been a body of work that has been going on in my lab for over 10 years, and we've helped contribute to particularly the development of the BET bromodomain inhibitors. When I started working on this over 10 years ago, there was less than 100 papers published on BRD2, 3, or 4. That has changed remarkably over the last few years. And you'd think as a consequence of that, our knowledge and the ability to answer many of the questions related to these key transcriptional coactivators would have evolved as well. And that certainly has happened. But I want to highlight here what we don't know about these key transcriptional coactivators. The first thing we don't know is why are essentially BRD2, 3, and 4 ubiquitously expressed. These have incredibly similar activity. They all work as transcriptional coactivators. They have similar domain structures, yet all of them are ubiquitously. Why do these proteins have 2 bromodomains? They have an amino-terminal bromodomain and a C-terminal bromodomain. And these bromodomains, they differ, BD1 and BD2 differ from each other by just a few amino acids, and they are conserved all the way down through phylogeny. And the reason for trying to answer this question is to understand whether selective bromodomain inhibition would be important. Every single small molecule that has been developed against a BET bromodomain inhibitor, that are BET bromodomain inhibitors thus far, equally inhibit both BD1 and BD2. They show no preference in the clinical context. We really got into this several years ago when we started to develop methodologies to try and identify how small molecules work on chromatin. Here we developed a method called click sequencing, where we were able to functionalize the small molecule and localize where it is able to engage its target. So here's the small molecule binding BRD4 whilst it's at chromatin. What this really showed us was that the reason we were able to do this is that BRD4 uses its first bromodomain, BD1, to bind chromatin. Its second bromodomain remains free to engage other proteins. And here we were able to localize where BRD4 is bound because the drug was able to bind here. We showed that it was the first bromodomain that was really important using genetics because we were able to rescue, loss of BRD4 with wild type BRD4, and also BRD4 that contains a mutation that abrogated the activity of the second bromodomain. However, BRD4 that contains a mutation that abrogates the activity of the first bromodomain was unable to fully rescue these cells. So again, providing functional evidence that it's the first bromodomain that was really important in a malignant context. And so this provides and finds domain-selective inhibitors of B1 and B2. And this is work that was led in my lab by Omar Gillen, a wonderful postdoc in the lab. And it involved collaboration across multiple different countries, including with GSK and Rad Prinja IMNA, and also in Germany with Paola Grandi and her group out of Heidelberg. In short, we were able to develop small molecule inhibitors that showed selectivity. So here is our previous BET bromodomain inhibitors that bound both BD1 and BD2 with equal potency. We developed IBET BD1, which was able to selectively just engage the first bromodomain of BD1 of the BET proteins, and IBET BD2, which was able to selectively engage only the second bromodomain. We showed the specificity of this using a number of different assays. And as part of this process, that previous small molecules that had been developed that have been purported to have specificity actually in vivo, in a cellular context, equally engage both BD1 and BD2. So using these small molecule inhibitors, we were able to recapitulate the genetic phenotypes that we had described several years earlier. Here what you see again is that small molecule inhibition of just the first bromodomain phenocopies the effects of the pan-BET inhibitor, whereas bromodomain inhibition of the second bromodomain fails to induce cell cycle arrest, apoptosis, etc. in both AML, breast cancer cells, and a number of other different cell lines. Here's the example showing you that there is no induction of apoptosis, there is no induction of a cell cycle arrest, and there's modest activity in both in vitro and in vivo against patient samples as well as mouse models of this disease. This then led us to really understand the fact that in many ways, small molecule inhibitors to just the first domain of BD1, of the first bromodomain of the BET proteins can recapitulate many of the effects that we'd seen, both clinically and preclinically, than with the pan-BET inhibitors. And this is because it results in a similar transcriptional inhibition of the major genes driving the malignant process. As you can see here, here is BD1 inhibitor, here is the pan-BET inhibitor. The genes that decrease as a result of these small molecules are virtually identical. Importantly, and rather surprising to us, is the fact that IBET BD2, the highly potent inhibitor of the second bromodomain, not result in any major transcriptional change at baseline compared to vehicle. And the reason for this, we were able to show, is that IBET BD2 is just simply unable to displace BRD4, BRD3, or BRD2 from where it is bound at chromatin. And here I'm just showing you the example of the well-studied mixed super enhancer, where BRD4 is bound and completely displaced by either the first bromodomain inhibitor or pan-bromodomain inhibitor. And this really occurs both at typical enhancers, but also at super enhancers, and occurs for all of the BET proteins. So what is the process? Why is it that the second bromodomain is even there? And more importantly, why is it even conserved? And really, to understand this, we went back to the fact that we have been looking at transcriptional inhibition of genes that are normally cancer cells. But what about gene induction in the context of immunoinflammation? And here, when we now start to look at the induction of genes, so here's an example where we induce the expression of class 1 on the cell surface using interferon. Again, here, the pan-BET bromodomain inhibitor stops this induction, as does the first bromodomain inhibitor. But importantly, for the first time, we started to see phenotypes with inhibition of the second bromodomain inhibitor. And here, what we can see is that the second bromodomain is required for transcriptional induction of the genes. Using nascent RNA sequencing, a method that was pioneered by a colleague of mine, Johannes Zuber in Vienna, we were able to show that transcriptional induction of interferon responsive genes was highly dependent on the second bromodomain. So here's all the genes marked in orange that go up in expression of interferon, and inhibits these genes, as does the BD1 inhibitor, but so does the BD2 inhibitor. Now, how does this work at chromatin? With transcriptional induction, we get loading of BRD2, 3, and 4 at these genes that require transcriptional activation. The pan-BET inhibitor stops the loading of these genes. The first bromodomain inhibitor does the same. But importantly, the second bromodomain inhibitor also now reduces the loading of BRD2, 3, and 4. Importantly, what I want you to see here is that the effects are far more pronounced on BRD2 versus 3 and 4. So to now understand why it is that all 3 BET proteins are expressed, we use an auxin-inducible degron to phenocopy the effects of small molecule inhibition of just BRD4. Because thus far, the field has mainly studied the transcriptional effects of BRD4. BRD4, we see a decrease in cell survival, as we have done for a number of years using genetics. Here we can see it with a small molecule. But importantly, here's the transcriptional induction with interferon of class 1, and here is the inhibition of this transcriptional induction with the pan-BET inhibitor. In contrast, just BRD4 inhibition is not able to effectively reduce the transcriptional induction, suggesting that 2 and 3 also play a major role in inducing the expression of class 1. And this again is shown here by RNA expression. Although genes that are just normally maintained in cells are equally affected by loss of just BRD4 compared to the pan-BET inhibitor, transcriptional induction is far more potent with the pan-BET inhibitor than compared to just loss of BRD4. How does this work out in immunoinflammation? Here again, now with a specific inhibitor, we do not see any effects on cellular proliferation compared to BD1 or pan-BET, but the induction of cytokines required for immunoinflammation are equally well affected. We did several models, in vivo models of immunoinflammation, and here is essentially what is my second last slide, showing you that with a very selective BD2 inhibitor, using an induced model of psoriasis that arises as a consequence of immunoinflammation, when compared to the FDA-approved phosphodiesterase inhibitor called apremilast, the BD2 inhibitor actually improves the psoriatic phenotype far better by decreasing the clinical score, decreasing epidermal thickness, and the reason that it is better than the FDA-approved drug is that it targets the source, which is the inflammatory cytokines that drive this process. It reduces the expression of these inflammatory cytokines. This process pretty quickly. This paper was recently published, so I'd encourage you to go and see it. And it really, for the first time, helps us understand why there are 3 ubiquitously expressed BET proteins, because BRD2 and BRD3 are also required for the activation of genes, whereas BRD4 is the main BET protein required for the maintenance of genes. We then developed small molecule inhibitors that essentially decrease just the maintenance of gene expression by inhibiting the first bromine domain and reduce the induction of gene expression by inhibiting the second bromine domain. BRD4, as I showed you, may be the main protein required for the maintenance of gene expression, but all the ubiquitously expressed BET proteins are required for effective gene induction. The consequence of this work that B1 is efficacious to pan-BET inhibitors as an anti-cancer therapy, but B2 inhibitors, although it does not affect maintenance of gene expression, may be very valuable in the context of immunoinflammatory diseases. So I'm going to stop there. There's a number of people to thank. This is the lab, as Sarah Jane and I run two separate labs, but we largely work as one big group. There is, a huge degree of diversity, both in the countries of origin of these people, but also their skill set. And, it's an absolute delight to work with them on a daily basis. I'm also going to shortly show you, put up the people to thank. This work that I've talked to you about today was as a result of collaboration from a number of people, both locally and around the world. And there are also a number of funding agencies, as shown on this slide, that really support our work and enables it to proceed. Particularly, I'd like the Cancer Council of Victoria, who provide me with my major fellowship, as well as the Howard Hughes Medical Institute, of which I'm an international research scholar, and also provides me with substantial funding. So I'm going to stop there and hopefully have some time for questions.
Wow. Mark, thank you very much. That was a tour de force. I invite folks that if you have questions to enter them into the Q&A section, and we'll start to go through those. Mark, I found that to be a really fascinating walk through the role of plasticity and targeting the epigenome in terms of a variety of malignancies. And it's interesting that for a lot of the drugs that I think about and the mechanisms of acquired resistance, they're very much associated with events that occur in DNA. So reversion mutations for BRCA deficient tumors, some of the genomic events that we see in HLA or B2M in solid tumors, but in fact, you really brought that home around the actual cell state and the cell of origin being a key point there, which I hadn't really considered. So I found that quite thought-provoking. Can I just start by asking you a question that given how crafty cancers are, and you've shown some quite compelling work there around the likes of the BET inhibitors, what can you tell us about acquired resistance to the likes of BET inhibitors? So once you play this epigenetic, you exploit this epigenetic vulnerability, I'm curious to understand, what do we know about how cancers try to become resistant to that?
Yeah, so I guess that's probably one area where we have studied in quite a lot of detail. And I guess in some ways, in contrast to say small molecule inhibitors of tyrosine kinases, where, for a long time, we know that you get these gatekeeper mutations that just reinstate the catalytic activity of the target, but render the drug relatively useless. In epigenetic therapies, there have been few, if any, examples where mutations do this. Because in many ways, the mutations would just phenocopy the effects of the drug. And, the critical amino acids that are required to bind acetyl lysine are essentially pretty similar to the critical amino acids required to bind the drug. So if you mutate these, you're essentially phenocopying the effect. The cancer cell has to adapt in a very different way. Instead of this being driven by a mutational process, it is driven by transcriptional plasticity. So it just finds another transcriptional coactivator to use when it can't use a BET protein, when it can't use a histone acetyl transferase. when it can't use a histone methyltransferase. It just takes another family member or another protein that does a similar job and instead uses that. With the BET proteins, what we've mainly seen is this process of enhancer switching or new enhancers popping up. And these new enhancers give rise to new transcriptional pathways being activated that ultimately still provide the same drive for the cancer cell. That is by reinforcing the expression of these key oncogenes such as MYC, BCL2, etc. Now, I think that that is a really important adaptive process in epigenetic therapies, but I suspect it goes beyond epigenetic therapies because the time sequence required to develop mutations, even I guess, if we think about, say, the BRCA reversions, Sean, you know, some of these patients who are manifesting what looks like BRCA reversions, that happens in months to a year or so. Yet we know that, you know, for a cancer cell to really acquire that mutation is probably that perfect mutation or that reversion, that's probably, you know, a process that takes, in some cases, substantially longer. So I guess one thing that we've been trying to understand is whether these cells are always pre-existing, right? And instead, what's happening here is that in the context of, say, PARP inhibitors or platins, that essentially the cells that are BRCA wild type have a greater clonal fitness, but these cells always existed in the first place. So that's one thing we're trying to understand, is whether these are pre-existing cells that just did not have the fitness to survive in the context of therapy, and that emerge only in the context of therapy, I should say.
Yeah, interesting. I must admit that the types of things like BRCA reversions, given that the drugs that we use, especially PARP inhibitors, are really promoting, I mean, they kill by hypermutation.
Yeah.
You're inviting chaos, aren't you? In many respects.
Yeah, absolutely, absolutely.
And I was curious whether there is an accumulation of other oncogenes or other genetic events that, rather than the enhancers, that might potentially be seen to be giving a different proliferative signal. But it makes perfect sense that if they can exploit a different epigenetic landscape to drive the genes that have been silenced, then that's an interesting mechanism. We have a quick question here from Fred. Fred has said, fantastic talk as always. He's curious to know what the impact of epigenetic regulating drugs, such as beta inhibitors, have on adult stem cells.
That's a good question, Fred. You know, the short answer is I don't think we know, right? We have not treated humans for long enough. We've not treated animal models for long enough to understand what the consequences are on normal versus cancer stem cells. It is very clear that many, if not most, epigenetic therapies do not eradicate cancer stem cells. That is highlighted by the fact that in those therapies that were able to induce that short-lived, and essentially the tumor reemerged. So eradicating the cancer-initiating cells, for want of a better word, just simply did not happen. And so there is no doubt that epigenetic therapies need to be used in combination going forward. What the cumulative toxicity would be if we add an epigenetic therapy with, say, conventional chemotherapies or other targeted therapies on cancer stem cells or normal stem cells, I don't know. What we know is thus far, you know, even the patients who've been treated, say, for instance, with BET inhibitor, we've treated a couple of patients for 2 years at our center. None of them really had any enduring problems with hematopoiesis or any other tissue, given the fact that this was a systemic treatment, but it was never combined. So it'd be interesting to see what happens in the context of combination.
Good stuff. I've got another question just to follow up, because thinking about the majority of the work that we've had going on at the centre is very much focused on solid tumours. And certainly a common theme running through a number of the cancer types we look at is that epigenetic regulators as a class of driver genes are some of the most commonly mutated whether it be SNF or the Compass complex, we're seeing them being mutated a lot. Do you have a feel as to those sorts of things there? Do you think that's just opening up plasticity, or are we driving, I don't know, canalization into new states there? And why aren't we exploring that more?
Yeah, 100% we think it's exactly that, right? You know, it provides, so if you think about SWI/SNF, apart from P53, there is, you know, SWI/SNF mutations occur in 20% of all cancers.
Right.
And, you know, they are not great oncogenic outside of the context of some pediatric cancers, where they clearly are major oncogenic drivers. But by and large, mutations in the SWI/SNF complex aren't really major oncogenic drivers. So they have to be facilitators. And so we had a paper last year, in 2019, it was published in Nature Medicine, looking at SWI/SNF mutations in the context of mantle cell lymphoma. And what we found there is that the SWI/SNF mutations, so SWI/SNF, for those who don't know amongst the audience, is a key chromatin remodeling complex, conserved all the way through phylogeny, and essentially also works as a transcriptional coactivator, moving nucleosomes around. And these mutations in the SWI/SNF complex, what we found is that it creates this perfect milieu that we term transcriptional noise. What we mean by transcriptional noise, that means an isogenic population of cells had multiple different transcriptional programs that increase transcriptional heterogeneity. And why is this the perfect milieu? Because you can imagine that any bottleneck applied will always have at least some population of cells with the right transcriptional program on to meet that bottleneck, right? And so that's the theory we put forward in that paper. And subsequently, there's been two Nature Genetics papers, one from Jason Carroll's group in Cambridge and another from the US, really showing that, you know, SWI/SNF mutations also confer, you know, increased resistance properties to anti-estrogen therapies, you know, and by enabling this plasticity. And so we think that that is likely to be a major reason why chromatin regulators, not just chromatin regulators, are a major target for mutational processes in cancer, is because it increases the opportunity for adaptation and noise.
Fantastic. Look, I'm mindful that we're coming up on time here today, so we might call that a wrap. It just leaves me to thank Mark once again for a fantastic presentation.